Welcome to another episode of Nick Noss, the Science Boss. Today we have our guest, Osvaldo. Hi, Osvaldo.
Hello.
So, can you tell us a little bit about fermentation?
Absolutely. All right. There are two ways organisms convert carbohydrates into energy: cellular respiration,
Right?
and fermentation.
Right.
Today we'll be talking about fermentation.
Right?
When oxygen is not available, most plants and/or fungi will switch from cellular respiration to alcoholic fermentation.
Right.
Fermentation is also known as anaerobic respiration.
Right.
This means that there is no oxygen.
Right.
Fermentation uses substrate-level phosphorylation
Right.
instead of electron transport chain to generate ATP.
Right.
In alcoholic fermentation,
Right?
pyruvate is converted to ethanol in two steps. The
Right.
first step releasing CO2.
Right.
Alcoholic fermentation by yeast is used in brewing, winemaking, and baking.
Right.
Humans also use a form of fermentation.
Right.
Our muscle cells use lactic acid fermentation to generate ATP when O2 is scarce. For example, when we work out, our legs may become sore. This is lactic acid releasing.
Right.
In today's lab, we'll be focusing mainly on alcoholic fermentation.
Right.
In today's lab, we need grape juice, yeast, sugar, water, and for you guys in the lab, a graduated cylinder, a balance, 2 beakers, a heat source, and a funnel. We're going to do this experiment on a larger scale, so our measurement's going to be different from your guys'.
For step one, what we're going to do is we're going to take some warm water that we had boiling earlier and we let it cool down to about 90 degrees Celsius. We're going to put that in our beaker. After that, we're going to have to add our 2 grams of yeast. We're going to give that a stir. And once we have that in there, we're going to let that sit for about 2 hours to let the yeast activate.
Alright, in step 2, we're going to be adding 300 milliliters of water and the equivalent part in the sugar. We're going to go ahead and put it in this pot, in your heat source. Alright. Oops. We're going to go ahead and stir that until the sugar dissolves.
For the third step in our experiment, we're going to measure out our grape juice. You're going to need 200 milliliters, and we're going to add that to the beaker. Once you have your 200 milliliters of grape juice, you're going to go ahead and dump in your activated yeast. And give that a stir.
Alright, this is step 4. This step we're going to add our sugar water mixture into our grape juice activated yeast mixture. Alright, we're going to go ahead and do that slowly. We're going to go ahead and stir this. We're going to go ahead and pour this into our pitcher, make it a little easier to put it in our jug. Alright, now that we have it in this pitcher, we're going to go ahead and put it in our jug.
Once you have your grape juice mixture in the beaker, seal it with saran wrap and poke a small hole in it so the gas can slowly release. Let it sit this way for 18 days to allow fermentation. Fermentation time, come on! Fermentation time, come on! It has now been 18 days since the start of our fermentation process. The wine has been filtered and is ready for consumption. Oh, Osvaldo, the legs on this wine are tremendous.
And the nose is superb.
I do agree. Fermentation time, come on! You're an idiot.